Good morning everyone. I am Dr. D. AkilaDevi, working as a professor and head in Department of Pharmaceutics, School of Pharmaceutical Sciences, Wales Institute of Science, Technology and Advanced Studies. Today I am going to discuss a new topic on novel drug delivery systems from Unit 1, Polymers, Classification, Structure and Applications. What is a polymer? The word polymer originates from Greek words poly means many and mer means parts. Definition: polymers are high molecular weight macromolecules made up of large number of simple repeated chemical units called as the monomers. These units are covalently bonded together in long chains resembling beads on a string. While small molecules might consist of a few atoms, polymers contain hundreds to hundreds of thousands of atoms, giving them entirely unique physical properties. Classification by source. The polymers are classified according to the source: natural polymers, synthetic polymers and semi-synthetic polymers. Polymers isolated directly from the natural materials in plants or animals. Example includes cotton, wool, silk, DNA, proteins and natural rubber. They are inherently biodegradable and non-toxic. Synthetic polymers. They are man-made polymers synthesized in a laboratory or factory, usually from petroleum-based chemicals. Examples include polyethylene, polyvinyl chloride, nylon and polystyrene. They offer engineered properties. Semi-synthetic. They are derived from naturally occurring polymers through chemical modification to enhance their properties. The primary example for this is rayon and vulcanized rubber. Classification by structure. The polymers are classified according to their structure as linear polymers, branched polymers and network and cross-linked polymers. Linear polymers. They are long continuous chains with no branches. They are packed closely together, resulting in high density and high tensile strength. Example: high density polyethylene. Branched polymers. Linear chains with side branches. Branches reduce the packing efficiency, leading to lower density and softer materials. Example: low density polyethylene. Network or cross-linked polymer. Chains bonded together by strong covalent bonds in 3D dimensional structure. Example: rubber. Thermal classification. Based on thermal classification, polymers are classified as thermoplastics and thermosetting plastics. Plastics that become moldable above a specific temperature and solidify upon cooling. This process is fully reversible. It is composed of linear or slightly branched chains held together by weak intermolecular forces or van der Waals forces. They can be reshaped, recycled and melted repeatedly. Examples are polyethylene, polyvinyl chloride, polystyrene and Teflon. Thermosetting plastics. Plastics that irreversibly harden when heated or treated with a catalyst, taking on a permanent shape. They are composed of heavily cross-linked 3D networks. They have strong covalent bonds formed between the chains during curing. They cannot be melted or reshaped or if overheated. Examples are epoxy resins, bakelite and polyurethane. Polymerization mechanisms. Addition polymerization. So it is a simple chain growth process, and the monomer requirement, they have multiple bonds, that is C double bond C, and the byproducts, they don't have byproducts, and the polymer molecular weight is integral multiple of the monomer, and typical products are polyethylene, polyvinyl chloride, polystyrene, etc. And polymers formed by condensation polymerization, it is a stepwise growth process. So they have two or more functional groups, and the small molecules such as water and hydrochloric acid are eliminated by this reaction. Not an integral multiple of the monomer is required. Examples: nylon, polyesters and polycarbonates. Properties of polymers. Key physical and chemical properties. Low density and high strength to weight ratio. Polymers are significantly lighter than metals or ceramics, making them highly desirable for aerospace, automotive and packaging industries. Chemical and corrosion resistance. Unlike metals, most polymers do not rust or corrode. They possess high resistance to harsh chemicals, acids and environmental degradation. Thermal and chemical insulation. Polymers are generally lack free electrons, making them excellent insulators. They are fundamental in casing electronic circuits and electronic wiring. Malleability and processability. Through heat and pressure, thermoplastic polymers can be molded, extruded, or cast into virtually any complex shape efficiently and economically. Polymers as pharmaceutical excipients. So they enhance the mechanical strength of solid dosage forms and ensure uniform disintegration upon ingestion. So they are used as binders and disintegrants. So viscosity modifiers. They control the rheological behavior in liquid suspensions, syrups and ophthalmic drops. Taste masking and film castings. They protect active pharmaceutical ingredients from moisture and mask unpleasant flavors. Solubility enhancers. They improve the bioavailability of poorly water-soluble drug molecules via complexation. So control drug and sustained drug delivery. Diffusion control. The drug dispersed through a non-porous polymer matrix or reservoir membrane at a consistent, predictable rate over prolonged periods. Swelling and erosion. Hydrophilic polymers absorb physiological fluids, swell in hydrogels and gradually erode to release therapeutic payload safely. Osmotic pumps. They are semi-permeable polymeric membranes utilize the osmotic pressure gradients to push the active ingredients out at a zero order release rate. Smart polymers in nanomedicine. Advanced polymer chemistry enables the creation of stimuli-responsive carriers that react to physiological microenvironments like pH drops or thermal shifts. These smart nanocarriers maximize therapeutic efficacy at diseased states such as solid tumor tissues while minimizing systemic cytotoxicity and healthy cell exposure. Advanced therapeutic horizons. Gene delivery vectors, cationic polymers, compact nucleic acids and DNA plasmids to traverse cellular membranes safely during gene therapy. Polymer protein conjugates. Covalent attachment of polyethylene glycol shields protein therapeutics from enzymatic degradation and renal clearance. Nanovaccine adjuvants. Polymeric microparticles prolong antigen presentation and stimulate robust, sustained and immunological response.